This is Blood and Cancer, the official podcast of MDedge Hematology/Oncology, where we discuss the latest research, treatment considerations, and other issues related to hematology and oncology. Welcome to this program. I'm your host, Dr. David Henry. You're listening to our weekly podcast entitled Blood and Cancer. Today, I'm really delighted to be speaking with Dr. Alice Chen, who's at the NCI, National Cancer Institute, in the Division of Cancer Therapy and Diagnosis, and she's the chief of the Early Clinical Trials Development Program. Dr. Chen, thank you very much for joining us today.
Dr. Henry, thank you for having me. I'm really excited to be here to talk about NCI-MATCH.
Well, I am just as excited as you, and it's a wonderful idea and program. So for our listeners, can you tell us what is the NCI-MATCH? How'd you come up with that acronym, and what's it all about?
So NCI-MATCH is a trial that is actually the acronym. It's been a while since I actually remember. It's the NCI Molecular Analysis for Therapy Choice. And basically that dictates what the trial is about. About 2013 or so, genomic medicine was starting to come into play in oncology, where specific mutations is a driver and that there are specific drugs that has led to great responses. Because of that, NCI started to look at the feasibility of trying to capture this new way to treat cancer. Instead of looking at lung cancer should be treated with these drugs, these are, tumors with this specific mutation should be considered for this drug that specifically targets that mutation. So we started building this trial 3-4 years before it actually opened in 2015. And one of the reasons we want to do it is with the NCI resources of having multiple sites through the NCTN and the NCORP, which are community sites, which then numbers over 1,000 participating sites, so that we're able to try to provide this trial to patients that may be in areas that are not immediately close to a large medical center, so they can stay at home to get treatment if possible. We also utilize some of our resources that allow us to work with pharmaceutical companies who has been great partners in providing agents for this trial. The trial ultimately is going to have 39 arms, though these arms had opened and closed through various parts of the trial itself. The original 6,000, when we had originally planned this in 2015, it was a normal practice to send biopsies for genetic analysis, though it is now. I think Medicare has changed that somewhere in 2017, which made it kind of a common practice. At that time, it wasn't, unless you are a specific histology. So we had actually biopsied the first 6,000 patients on the study. We had developed an assay to look for specific mutations for drugs that we are able to put onto this trial. So for the first 6,000 patients, they have to be biopsied, but that provided valuable information in this exploratory study because we were able to know what the mutation of the tumor is prior to treating the patient. So the difference there is that sometimes in the past and even now, the mutational analysis is done of the original tumor, and patients may have had several treatments since then. That may affect the changes of their molecular characterization of their mutations. For the first 6,000 patients, we really wanted to know, as close to the time we started their treatment, what the mutational status is. And about 38, we were very successful in analyzing the tissue. About 95% of the tissue were analyzable, and we were able to do analysis on those. 38% of the patients, we did find an actionable mutation, which is a mutation in which we want to target. However, in this study, because it is exploratory, we wanted to not look at information that's already available. So we excluded tumor from each of the arms if that particular drug is already known to be effective for that particular histology. So we may have excluded breast cancer, colon. For BRAF, we excluded colon. Melanoma, we excluded for the BRAF because we knew the data there. We wanted this trial to be exploratory, to provide interesting findings that we can take into further study. One of the other things we wanted to provide in this study is to look at rare tumor. You know, it's easy to do breast cancer, colon, because there's a large number of patients with it. However, for rare tumor patients, one, there's not a lot of study, and there's not a lot of study close to home for a lot of these patients. So we actually have slotted 25% of the slots in the original 6,000 for rare tumor patients. And it turned out 60% of the participants had what is deemed as rare tumor for this particular trial. We had defined the rare tumors, anything aside from breast, colon, prostate, and lung. So we were very excited about that part of the trial itself. The trial has opened in 2015. We opened only 10 arms at the time. We had closed the trial after 4 months to look at, you know, it was an interim analysis that was built in because we've never done a trial this large before with this many sites, and we wanted to make sure everything worked. One of the issues that came up was that we were having, because of the excitement of all the participants, there was a large number of patients that came in. It overwhelmed our laboratory system. And so we shut the study down, increased our laboratory support, opened additional arms. So there were 24 arms that opened at that when we reopened the trial. And ultimately we opened 30 arms by the time we finished the 6,000 patients. We did open 8 additional arms and has 1 more arm that is yet to open on this trial. However, in 2017, once we completed 6,000 patient biopsy, we have moved on because of the standard of care has changed. Mm-hmm. We have talked to a lot of the laboratory partners who were willing to work with us to identify patients with specific mutations for the extremely rare, what we think are more rare, the arms that have not closed yet at that point with a prevalence rate of probably less than 2%. And so after the 6,000 patients, we, instead of biopsying patients, we actually went to having laboratories help us find these patients. And we have completed probably 50% on the first 6,000. And ultimately we have completed additional arms off of using the laboratory recommendation method. So
well, that raised a few questions that for my mind and clinicians listening. Mm-hmm. So of course, NCI-MATCH, molecular analysis for therapy choice. So a really nice acronym. And I think you mentioned early on, you had biopsy specimens from original diagnosis, but they've been treated a lot. So for study patients, was the genomic analysis on a current biopsy or original biopsy? How did you decide there? And I gather no liquid biopsies, only tissue.
Only tissue. And actually for the 6,000, we biopsied patients right before treatment.
So you had a current analysis of whether they might have evolved.
Right.
And so again, to get my head around it, I'm a site in Iowa, let's say, and I'm a part of the trial, and I have a patient, I do a biopsy. Is that sent for central review for molecular analysis to NCI?
Yes, so we actually have a laboratory system set up for 4 labs around the country.
So I think you made it clear. So those analyses come back. You tried to avoid if someone's BRAF mutated melanoma, we know about that, we move on. Right. But we find mutations that have a drug in your pharmacy. And now again, I'm in Iowa and a central lab does this and I've got a genetic mutation, AKT3, and you have a drug for that. How do I get that drug out to my site and administer to the patient? So for the sites are all selected and they have to have the trial open at their site. They're part of, most of the community sites are part of the NCORP system, which is part of the NCI's, one of our network systems, which has things set up so that CTAP, the Cancer Therapy Evaluation Program of the NCI, their pharmaceutical management branch,
will send them the drug. So usually if a patient is identified, the site can order the drug and within 24 hours will ship the drug to them.
Wow. Okay. And then the drugs administered at the site. And before I forget, these are all, I'm sure, IRB approved sites. And some of our listeners may either want to become a site or find out where the sites are. So we'll put on our webpage, maybe get from you later, where all the sites are across the country for listeners. And if someone wanted to be a site, are you fully site accrued or are you interested in more potential sites?
So these sites are all part of the NCTN or the NCORP system. So if a site is interested, they would actually have to become
Part
part
of
of
that.
the NCORP, yes.
Okay, so we'll make it known to investigators throughout the country where it might be local to them to refer such a patient, because it's really exciting because you've done the mutation. This is what we're all about. We're just evolving in that area rapidly. You've found a mutation that might be actionable, drugs sent to the investigator, given to the patient. Now, how do you then evaluate? The patient is at that site, gets the drug. How do you evaluate going forward, win or lose, when to decide to stop or not?
So for patients that start on treatment, there's a baseline CT scan that's obtained, and we will continue to scan them every 2 cycles to see if they have at least stable disease or partial response or complete response per RECIST criteria.
Mhm.
For
All
those
right.
patients who progress, they'll come off study.
Okay. So we're now, it sounds like 5+ years into this. Can you share any results? You're cycling drugs in and out. They work, they continue, they progress, they come off. Do we know any drugs that might be moving up or down in your enthusiasm because they work or don't work? Anything, and I want to mention to our listeners, the study you co-authored all about the NCI-MATCH came out in the JCO in 2020. So we'll post that reference on our site as well. So anything you can share about how responses, good, bad drugs went?
So the responses have varied in this study. And the one thing I should mention is that for this particular study, we had originally designed it with monotherapy. And we're fully aware that for monotherapy may not be the best path forward for a lot of these targeted therapy trials. The first one to report is actually nivolumab. Immunotherapy, very exciting. So that was reported in JCO. Subsequently, various other manuscripts has been published for currently I think there's been 5 arms that's been published.
Okay.
Response rates vary anywhere from 2 to 38%. So, it's one thing to keep in mind is the patients that are accrued to this particular trial are patients that has gone through all known treatments, standard care for their treatment. So to some extent, these are really patients that potentially is considered for phase 1, phase 2 trials. So, even though there's a few responses in some of the arms, I think even those are extremely exciting in exploratory setting, especially if they have a rare tumor to find that information. And there is a network that's set up. We're continually monitoring the accrual to these arms. We constantly look at the data so that we can look to see if the information is mature enough for reporting, so that we try to get the reports out either through an abstract format or manuscript as soon as possible. Because I think that is the one thing that is important in precision medicine, any of the common medicine, oncology these days, or things are moving so fast, you want to make sure that if you have information to share as soon as possible. And so that is a continual process for us.
Absolutely. And you mentioned just now in the paper that I mentioned the one from 2020 JCO, that maybe not monotherapy is enough, maybe combination therapy like a lot of oncology. Any results you might share there, or combinations of 2, it's probably 2, I would suspect not more, drugs that you are finding exciting versus not?
So for this particular trial, there were only, I believe, one arm that really came out to be a combination. And it's dabrafenib with trametinib that was reported as positive.
Yeah, the melanoma combination, yeah, which impresses in melanoma, and you found that in other where you have the
in
target.
other tumors.
Mhm.
Yes. So I mean, that is one of the arms that is exciting, but that is a combination. So, there is currently in the plans to look at combo match, which is to look at combinations of different targeted therapies to kind of bring the information. It's the next match. I think the next
Yeah.
logical step for us to go is that we know now some of the information for the single agent, for especially some of these rare tumors, to use that to help guide us into the combination trials, in terms of is there specific histologies that are interesting? Are there ones that we should avoid? So that is one of the things that are currently in development.
Well, the beauty, I'm listening to this and really getting enthusiastic. The beauty of this trial is you have a large network, you have genomic analysis changing over time. New mutations are being found, new drugs are coming along. So that keeps added to the pipeline that you're making available. Patients come on and off study quickly. If they progress or if they respond, they stay on. And then now you're moving on to combination, as we do in a lot of oncology, to see where those responses might be. Actually, I'm thinking of a patient I had with pancreas cancer a few years ago who had a BRAF mutation that did respond to that combination you just mentioned, melanoma. So that was my N of 1 and yours is N of 1,000. So it's really exciting. Well, anything you can share, and we'll post these publications on our website so listeners can review. Anything you might share of where the latest updates are of what you're doing next? Any plans to change the trial? How's it going as of today, late December, I'm sorry, early January 2021?
With a new president.
Yeah, right. Thank you. Our first one with a new administration, yes.
No, I think that for NCI-MATCH, we are in the process of trying to complete some of the rare arms. And, there is still a lot of work to be done in terms of the back end. I think one of the things you brought up had been that we did the biopsy in the first 6,000 prior to treatment, but we are in the process of trying to get archival tissue for those patients so that we could compare their original diagnosis versus the biopsy that we have done. So there are still a lot of genetic analysis and other things we're looking at in the current NCI-MATCH trial. Additional information can be found on the ECOG website, which we can forward you the link, which will tell you what are currently the available arms that are open. And there are 9 arms that are still currently open with 1 yet to open. So there'll be 10 open arms still. The one thing I do need to mention is that NCI-MATCH now no longer biopsy patients. So we are using either Foundation, Caris,
Some of the commercial labs.
some of the commercial labs now are helping us identify patients because it is so common nowadays for most of the patients at the time of diagnosis or recurrence to have that sent. So we've decided to utilize that resource instead, and we'll send you the link to all the labs that are involved as well. So, it's giving all the labs proper recognition.
Of course. So, and not liquid biopsy yet, although I suspect you might go there one day.
We might, so for NCI-MATCH, we are in the process of closing down the NCI-MATCH in terms of accrual. There's, as I mentioned, there's still a lot of work going on, a lot of interesting data, hopefully that'll be developed. But as for the next precision medicine trial, there are other, we're planning the combo match, and we're looking at immunotherapy as well. So for NCI, there are currently in development various other trials to continue what was found based on NCI-MATCH.
Well, this is wonderful, Dr. Chen. I can't thank you enough. I want to be respectful of your time and our listeners. That's more than my head can handle in one 20-minute session, but I think I've got it. And I want to thank you again. We've been speaking to Dr. Alice Chen is at the National Cancer Institute, Division of Cancer Treatment and Diagnostics, and where she's the chief of the Early Clinical Trials Development Program. Dr. Chen, thank you very much for a really informative session.
Thank you, Dr. Henry, for inviting me and giving me the chance to talk about something I really love and I'm very excited about. So
it comes right through. So I really appreciate it and good luck on your work. Blood and Cancer is produced by MDedge. Our editor is Jen Smith. All MDedge podcasts are produced by executive editor Kathy Scarbeck, and I'm your host, Dr. David Henry.